Canada approval suggests that this has been looked at and it is safe. It has to be efficacious. It has to conform to certain manufacturing standards, and they have to conclude that the benefits of taking the drug far outweighs the risk. Then they issue a notice of compliance, which suggests that they have gone through all these processes and they've given approval that the drug should be marketed in Canada. And then they issue a drug identification number. So for a drug to become available for sickle cell in Canada, Health Canada has to approve it. Next slide. But that's not the only thing. It goes through what is described as a Patent Medicine Prices Review Board. This is a board that has been established that regulates the prices of patented medicines sold in Canada. It is an independent, quasi-judicial body, and one of the 5 health portfolios within the federal government. So the other agencies are listed there for you, including Health Canada, Canadian Food Inspection Agency, the CIHR that oversees research, and the Public Health Agency of Canada. So the PRB is supposed to be one of these health portfolios of the federal government. It's supposed to work at arm's length from the Minister of Health and determines the maximum price for this patented drug. But currently, all of us should know, and all advocacy groups should know, that there are ongoing guidelines to implement a new patent medicines regulation that probably could slow down significantly the introduction and the launching of new drugs in Canada. Next one, please. That's not enough. There's a Canadian Agency for Drugs and Technologies in Health. This agency provides objective evidence to inform optimal use of health technologies, which includes drugs, diagnostic tests, and medical surgical devices. Supposed to be independent again and not for profit, and decides public reimbursement processes within the country. So this agency is independent and works on its own. Next, please. Then there's the Pan-Canadian Pharmaceutical Alliance, the PCPA. This body manages access to clinically effective and affordable treatments. So affordable has been highlighted there. It's an alliance of federal, provincial, and territorial bodies and conducts joint negotiations for brand name and generic drugs. This body, the PCPA, oversees federal, provincial, and territorial drug price negotiations. Next, please. This is a very multi-layered process. As you can see, it's multi-layered. You have a number of independent bodies. There's no clear indication of how they talk to each other. The process could become very cumbersome and could be a long process, a long winding process. But what is even more troubling is that the new guidelines that are being discussed could be a long winding process that would be a disincentive for drug developers to launch their products. So I've been in touch with the developers of voxelatol and crizanlizumab. And currently I can tell you on the list of countries where they are going to launch, Canada hasn't even showed up because there's the belief that the process for launching in Canada has been made more complicated, has been slowed down, and probably also may force them to lower prices at a level that they are not ready to do. And this is an important issue that came to a head with this article in the Toronto Star, that Canada is at risk of losing world-class medical treatments because of this cumbersome process. The overarching troubling thing about this is that the process doesn't seem to be transparent. It doesn't allow advocacy groups like Sickle the Rare Blood Disorders Group in Canada, and other agencies that are seeking to make sure that expensive drugs are made accessible to Canadian patients. It doesn't provide enough transparency. And I think this is a time for us to really have a strategy to make our voices heard, because there's no use to have a drug developed when it is not accessible to Canadian patients. And my patients keep asking, when is crizanlizumab coming? When is voxelotor coming? They know the evidence shows efficacy. They know that it's been launched in the US. The European countries are launching it. And the frustration that I feel and my patients feel is how long it's taking to launch new medications in Canada. Next, please. But currently, these are the costs of sickle cell disease therapies in the US. I'm comparing hydroxyurea with the new drugs that have been developed. Generic hydroxyurea will cost about $1,200 annually per patient, compared with voxelotor, which is probably estimated to be $125,000 per patient. Crizanlizumab is around $100,000, and L-glutamate around $30,000 annually. My fear is that these costs, comparable to what it is in the US, might be considered too expensive in Canada and might go through a rigorous process, that multi-layered process that I've outlined, and this might delay the process of launching these medications in Canada. BMT, bone marrow transplant, is accessible, but it should be more accessible. And as we talk about gene therapy that you're going to hear about, the question we should be asking ourselves, when these products and these processes are deemed evident in terms of efficacy, in terms of safety, and are approved by the FDA of the US and the European Medicines Authority, how long will it take to launch in Canada? And that's the question we all have to ask. Next slide, please. So what about access to treatment in low resource settings? Newborn screening is still a problem in many low-income countries. In fact, there's no country yet in sub-Saharan Africa that has a universal newborn screening program for sickle cell. Many who should be on penicillin prophylaxis, which is affordable, are not on it because they've not been diagnosed with sickle cell disease. Thanks to the Gavi initiative, most countries now, particularly the poorest countries, have access to pneumococcal vaccines through primary immunization. Folic acid is affordable. The Gates Foundation has done a lot of work with malaria prevention programs, but hydroxyurea treatment is still a problem in terms of availability and affordability. Thankfully, there are some steps being taken. Oxyurea is being produced in Nigeria. And what is interesting to note is they have a 100 milligram formulation, a 250 milligram formulation, and a 500 milligram formulation, which we cannot even have in Canada. We only have a 500 milligram formulation, which makes it extremely difficult when you are dosing for children. So they are manufacturing all these formulations of hydroxyurea. But even though it's being manufactured internally in the country, it is still not affordable to many families who are low-income earners. And that is a challenge for many countries in sub-Saharan Africa. Access to chronic blood transfusion is not possible because of inadequate blood supply and also the affordability of chelation therapies, which can be way beyond the resources of many of these systems and families with people who live with sickle cell disease. Next, please. Now, this is to show you that hydroxyurea has been studied in Africa. It's been proven to be effective and safe, and it's been shown that actually the higher the dose you give to these children, the better their response and the better they are in terms of complications with acute chest syndrome and pain crisis and need for blood transfusions. Next, please. So what is being done about this? There's a global coalition to combat sickle cell disease that's being co-chaired by the U.S. Health and Human Services Department, the World Bank, and the World Health Organization. And it consists of multiple organizations that are coming together to try to make some of these therapies and services accessible to low-income countries, such as newborn screening, early diagnosis, vaccinations, and access to hydroxyurea therapy. We are hoping that with the World Bank involvement, that many of these countries will receive the financial help they need in structures that enable them to sustainably provide hydroxyurea to people who live with sickle cell disease in low-income countries. So in summary, drug developers are doing their work. Many more drugs are available to manage sickle cell disease, but as yet, Canadian patients are not having access. If we do not advocate, if we do not make our voices heard to make sure that the multi-layered process is made more transparent and our voices are heard, I worry that it may take a long time before Canadian patients benefit from these developments that have taken place with treating sickle cell disease. I will stop here and invite some comments and questions.
Thank you so much, Dr. Odami, for this insightful and very informative presentation. We do have one question in the chat from Erin Colmetz. And so her question is, would national pharmacare increase or decrease access to new drugs and therapies?
That is yes and no. I think if a national pharmacare is going to be a smooth process that is not multi-layered, I think the multi-layer process actually makes it more complicated. Countries like Germany and other European countries have simplified the same process. Can you imagine that after Health Canada approval, all we needed was one body to look at it, look at the indications, look at the cost, be able to negotiate the price with the developers on a national scale, and then quickly come to a reasonably price for these drugs. I think there's also a comparison that they have introduced with many other countries. So there are, I think, about 12 or so countries that Canada compares itself with. So it looks at prices in Australia, in places like Sweden, in places like the UK, and other countries, and fixes the price. So there's a process, but that process should be smooth, it should be transparent, and the central goal of that process should be how quickly can Canadian patients have access to a drug that is proven efficacious and safe. So for example, I'll tell you that with the COVID vaccines, the government of Canada took a wise decision that it's not going to take COVID vaccines through that process. So there was a direct, once Health Canada approves it, emergency authorization, all these processes were cut out in order not to delay access to COVID vaccines. So I think that is the principle that should be driving this. And we as an advocacy group of patients, families, parents, professionals should be making sure that when it comes to sickle cell disease medications, the only one we've had is hydroxyurea for a long time. New ones are on the market, and we should make sure that patients get it the quickest. So rather than making costs drive the issue, we should make need and the speed of access the driving force behind it.
Thank you. And that's a great segue. We have time for one more question. And so Beverly Chung Brown has asked, are the new drugs treating sickle cell disease more efficacious than hydroxyurea and more beneficial for SS patients?
I think the process that this has to go through, if you have sickle cell disease and you have a high hemoglobin and you are not hemolyzing, breaking down your red cells so fast, I don't think you will need voxelotor. Secondly, if you have sickle cell disease and hydroxyurea has virtually controlled your pain, you hardly have any pain crisis, you won't need crizanlizumab. So they are there to be supplements. In fact, in all the clinical trials, some of us who have participated or in their design or in advising the developers have made a case that they should study the drug both alone and in combination with hydroxyurea. Because when I showed you the cost of hydroxyurea, you can hardly find a drug so cheap, just $1,200 annually, so easy to take once a day orally, that has that level of efficacy. So these drugs are not going to replace hydroxyurea. I think what they are going to do is to help patients who, despite hydroxyurea, or for one reason or the other, do not want to take hydroxyurea, that these patients will benefit from these other drugs. So not everyone will need it. Not everyone needs to be prescribed, but there are many patients today who still have a lot of pain episodes despite being on hydroxyurea. Remember, hydroxyurea reduces the pain generally if you take a whole population by about 50%. So there's still pain episodes, and crizanlizumab can be of help. Or they are on hydroxyurea, but they're still hemolyzing a lot. Their hemoglobins are low. They have anemia and fatigue. And we know that long-term breaking down of red cells can affect organs. In those settings, voxelotor, even in addition to hydroxyurea, will be indicated. So this is the process we have to take these drugs through so that there's a clear indication who needs it, but access to it should be quick and should be organized in a way that Canadian patients benefit from these new developments.
Thank you so much, Dr. Odami. There's some comments around the need for increased political advocacy, which you've spoken very well to in the chat. And then in the interest of time, we'll move on to our next presenter. Dr. Odami, I understand you're on call today. I don't know if you'll be able to be here for the Q&A afterward, but if Dr. Odami is there at that time, he'll be able to answer further questions.
Thank you.
Thank you, Dr. Rabena and Dr.
Excuse me, please, before Dr. Isaac leaves, please, I have a question for you, Dr. Isaac. I heard that you mentioned stroke prevention therapies. Please, can you shed more light on that? And what period is it best to be done?
Thank you, Yemisi, for your question. We are going to address further questions further along the line. We've gotten to the time for our next speaker, so we are going to introduce the next speaker. Your question will be answered at our question and answer session. Thank you. I would like to introduce our next speaker. Our next speaker is Donna Wall. Donna Wall will be speaking on gene therapy. He is currently leading the bone marrow, blood and marrow transplants stroke cellular therapy section at the Sickle at the University of Toronto. She has experience in transplantation for treatment of cancer, immune and hematological disorder. She is currently leading and is involved in a range of clinical trials and translational research in the hematopoietic stem cell transplant and cellular and gene therapy for the treatment of sickle cell anemia. And Dr. Wall holds the Garon Family Stroke Hats Off to Kids Chair in the Childhood Leukemia Research. Welcome, Donna Wall.
All right, thank you so much to the moderators and to the SCAGO team and to everybody who's given up their Saturday morning. It's truly a pleasure to give you an update of where we are with the very first experiences that we're having with gene therapy for sickle cell anemia. As you know, we know a whole lot about sickle cell disease and how there's one gene that a mistake is made that results in the red blood cells, instead of being these nice happy donuts, having very sharp edges on them, and that is the root cause of the disease process with the pain crises, the chest syndrome, etc. What I'm going to present to you today is hot off the press with our first experience using gene editing with to change a patient's own blood making, so they will preferentially make blood that is like that of a baby, and not express the sickle gene. And basically what that does is that it keeps the red blood cells with their healthy shape. This was recently published in one of our, the first experience in one of our more prominent journals. And then what I'm showing you today is from a presentation last month at European meetings for the update. So this is straight hot off the press. You're getting this information. You have as much information as anybody does here. So the idea here is that normally the kind of blood we make changes from before birth, right around, between time of birth, which is shown here on the bottom line, birth being 0 to 3 months of age. And you switch from making baby hemoglobin or fetal hemoglobin. And you're going to say, I've heard that before, because that's what hydroxyurea does. That gives me more fetal hemoglobin. And we switch to adult hemoglobin. And the problem with sickle cell is there's one little amino acid change that results in how the hemoglobin behaves in the red blood cells and triggers the whole set of problems. So we have right around 3 months of age, this transition happens. We do know that patients people walking around, not necessarily patients, walking around, there's a wide range of how much fetal hemoglobin people have. And there are actually families who just, based on their genetic makeup, only make fetal hemoglobin, and they have live normal lifespan. Normal lifespans, don't have trouble with carrying oxygen. So those are rare patients, but we do have some experience there. And we do know for population, at big population levels experience for patients who have sickle cell anemia, that the higher your fetal hemoglobin is, the less you have in the way of sickle cell complications. So the whole idea behind this first generation of gene therapy studies is to say, let's cut that switch. Let's prevent the switch from the body making fetal hemoglobin to adult hemoglobin. Let's just stay in the baby stage. And to do that, we took advantage of a really rich decades worth of research into how blood is made, how hemoglobin is made, what are the control points of hemoglobin, and what has been targeted is a gene that is a controlling gene or a transcription factor that says, hey, when I make this gene, and the body is reading the DNA to make to eventually make the protein that becomes the hemoglobin. That gene says, instead of making the fetal hemoglobin, switch and make the adult hemoglobin. Okay. And what we do with this first generation of gene therapy studies is we basically cut that switch. Okay. And so that so then the cell, as it makes new hemoglobin, is going to say, oh, I'm not getting the instructions to make the adult or sickle hemoglobin. I'm just getting the instructions to make the baby kind, which does not have the key here. It does not have the sickle defect in it. How we got at this scissors. I'm showing you scissors because this is exactly, we're really just doing a little electrician work here, and we're cutting some wires. Recently discovered is the finding of something called CRISPR-Cas9 system, and that is something that is so primitive in mankind. It's present in biology. It's present in yeast. It's present in bacteria. And basically it is a tool so that we can go in and it's evolved to protect ourselves from viruses and things that don't belong in our gene material. but we can specifically target just right at the sweet spot of the genetic material that makes this gene that does the switch, and we can cut it. Okay, so that's what you're going to hear me use the word CRISPR in Cas9. And it's incredibly specific in the way it does this cutting. The process, even though the system is straightforward, the idea is that we cut the switch and then we teach the patient's blood making system is only going to make baby hemoglobin for the lifespan. So it's a simple idea, but the process is still what I'm going to, I'm involved with it, so I'm allowed to say it. You're not allowed to say it. I'm just going to say it's clunky, and it's a big deal. And and how we do it is it's on clinical trials because it's new. It's the first time we're doing it in man. We once we find a patient who's well enough but is having a lot of trouble with their sickle cell, so we're looking for very special patients. We collect their young blood making cells. We ship them to a manufacturing site, collect out very young blood making cells, and so that we call them CD34 positive cells. We in the test tube we treat them, so we do that scissor activity. It's just done in a bag, and then it's all done. We're not putting in any genetic material. It's an enzyme. It's like we're sort of just doing a treatment. And so from then on, those young blood making cells, any blood that's made from them will be fetal hemoglobin. Okay. We freeze them down, we ship the cells back to us, we do a whole ton of testing to make sure that the cells are working, and then the patient's ready to get the treatment. We can't just give those corrected cells to the patient because inside their blood making system are regular sickle cells that are going to, there's no space for them to set up shop and make blood. And so we would basically, we need to cheat a little bit. We need to make some space so that these corrected young cells will cause blood making, and our goal is to cause blood making for life. So unfortunately, in 2021, right now, we have to give some chemotherapy. We give 4 days of a very strong chemotherapy drug. I'm not hiding that fact. And then once that medicine's peed off, we infuse the young blood making cells, and it takes about a month for the blood counts to recover. This is the most recent information on the first. This treatment is being done both for patients with thalassemia, which is a different hemoglobin problem, and sickle cell. So far, there have been 3 patients in the world with sickle cell who have been followed long enough to give report. So these are young adults, and they were patients who were really struggling with their sickle cell. They had an average of 7 vaso-occlusive crises a year. So, and that's despite everything that Dr. Odami and Simpson would be doing to prevent the sickle cell. It took roughly 3 weeks for the blood counts to recover after the treatment, and we learned a lot in these first few patients. It's a little hard for us to collect the young blood making cells. Sometimes we have to collect 2 and 3 times to get enough cells. But here's the results. And so just focus, don't worry about the numbers. Focus on the teal, the light blue coloring here. The top box here, this is the hemoglobin. And so these are the 3 patients. This is before they were treated, and you don't see any of this teal in them, okay? And after they were treated, this is some transfusions that they required. And then gradually over 3 months, and then longer as time goes by, about half the hemoglobin that's being made is fetal hemoglobin. You combine that, and basically what that is doing is it's making the patients are acting like sickle trait patients. So how are these patients doing? We're seeing fetal hemoglobin. So what this is months after treatment, and the three different colors are the three patients. And months after treatment, we're seeing in the blood, in each red blood cell, that there's fetal hemoglobin in each of them. So so it's not just in a few red blood cells, it's in all their red blood cells, which is really good, okay? Because what you want is some of that fetal hemoglobin to interrupt and prevent the that lining up of the hemoglobins that gives you those stiff red blood cells. And the good news is the hemoglobins in the patients have been excellent, in the 11 to 13 grams per deciliter range. But what's important is that they're no longer breaking down red blood cells. Okay, so one of these, this middle patient here, what used to be called yellow eyes, because he had such rapid breakdown of his red blood cells that he was always jaundiced, and he was so happy for his college photos that he could have a picture with white eyes in them. But so basically we're incredibly excited, but I'm going to just say, and what you should tell me is, okay, wait a minute, Dr. Wall, here, we're only at 18 months for the longest follow-up. What we do is we look to see, these patients have actually volunteered to have bone marrow tests done after treatment, and in follow-up, some of these are patients who have had thalassemia as well as sickle cell. We show that at the follow-up visits that the blood making continues to come from our corrected cells that we gave the patients, that it's sitting at 80%, 90% of the blood making is coming from the corrected cells. And what is going to happen, we're now, I've got, our longest follow-up patients out at about 18 months, and the others at a year. We're so far they're continuing to not need transfusions, and we're incredibly excited. But you should come back to me and say, but Dr. Wall, where's my 5-year and 10-year data? And we don't have it yet. Okay. This is a first example, first trial of a gene therapy, and there are many, many different strategies. We look at disrupting that BCL11 enhancer. So what we're doing in our trial is this circle, but there's many different correction approaches. And I can see in the next few years, that we will have the tools to, let's just get that S out of there. The different ways to do the blocking. We can add genes in, and so basically what we're describing now is first incredibly exciting, promising results, but we're still very much early stages. There also is the next generation of that CRISPR system, where we can actually engineer in the correction, not just cutting the wires, but doing the correction. And the part that I really dislike the most about this is needing to give the busulfan, needing to collect the cells. And so one of the bright guys in our laboratory is working hard at developing tools so that we won't have to pull the blood making cells out. So we can use, and this is, kind of, a dream project for us. We can deliver the scissors or this repair with by giving something in through the bloodstream, getting it so that it homes into where it needs to go. So just to say, this is exciting. But this is our first first experience. We are watching. We're doing a fair amount of meddling with Mother Nature. We're taking those young blood-making cells and we're changing them. The reason we went with the trial that we did, it does not include a virus. It does not include putting something in. But we don't know what happens when we work with the hematopoietic stem cell. So just to let you know, there's There has been a case of a patient who was treated, who did develop leukemia. That patient received a different gene therapy strategy that involved a virus with it, but there's no sign that that virus had anything. But when they took a look at, had an impact. When they took a look at the bone marrow that had been stored on the patient from before he had the treatment, they could already see that he had changes that were that went along with being pre-leukemic. So we do know we need to follow patients carefully. This is very much early stages. This is where I get all my my updates on my research, which is the New York Times, and who's been following this whole story very carefully, but basically saying that the treatment itself was not linked to the cancer, but we have to be very respectful. So the we are making, finally we're able to, I'm able to talk to your group and saying we're making progress. The first patients that we treat on study are going to be those who are sicker. So I know I'm talking to a lot of families who, yeah, everybody's interested in it, but it just, just to say it's not yet ready for patients who are doing well. And hydroxyurea, I've been incredibly impressed with how revolutionary it has been in sickle cell care, but there's still but we are, hopefully the field is moving very quickly. We have to make it safer. We have to make sure that it does cause lifelong correction. So at Sick Kids, our CRISPR trial has now accepting patients down to 12 years of age. To be eligible, you have to be really quite sick from your sickle cell and not have an immune matched sibling. We do have a second gene therapy study that should be opening this fall. And so I'd like to just end by saying this is a team effort, and thank you to Sickle, who's, we've been sharing this information for several years now, and getting out to the community about what the gene therapy is and isn't. We are collaborating with Drs. Ward and Koo at UHN and the Sickle team at Sick Kids, as well as our referring teams. We have a family within our bone marrow transplant team, but basically the whole Sickle Cell Sick Kids Hospital and Sickle community is involved with this program. So that's what I wanted to share with you today. And I have to be careful because I know I talk a lot, and I don't want to overtake my time.
No, thank you so much, Dr. Wall. This was an amazing talk, and it's amazing to see that Canada is on the leading edge of developing these curative therapies. So thank you so much. And so we have a flurry of questions in the chat, but there are some common themes. So I'm just going to try to narrow it down. So firstly, can you speak to natural ways or the ways that individuals with sickle cell disease are able to maintain higher levels of fetal hemoglobin? And then the other question, which you touched on, are related to who would be eligible for the trial at Sick Kids, and what are some of the risks that they have to know about in enrolling on this trial?
Sure. And so you can look things up on clinicaltrial.gov, and I'll get the SCAGO team the email link for that, and it runs through in detail, the requirements to be on the studies. How your body is, how your body is wired to make fetal hemoglobin. This is, probably better for Dr. Simpson and Dr. Odami. You're basically born with it. And some families and some people who live in different areas of the world just have higher fetal hemoglobin levels, and they tend to have less problems with their sickle cell disease. Your hydroxyurea is a wonderful tool to increase your fetal hemoglobin. And so that's, but to take, just watching, I've now had the privilege of, for the, 2 sickle cell patients that we're treating on the study, I've screened over 30 patients that have come to me pre-screened by the hematologist. Like some of the patients are falling off because they're too sick. Some of the patients are saying, some of the patients with sickle cell are saying, let me just wait a few years. Let me just see how this works. So it's kind of like, do you buy the, the most, current iPhone, or do you just kind of wait, and let things sort out and figure out which one works better. So it's, some of us are early adopters, and some of us, just a little bit more cautious. And then a lot of families I've talked to, and they're just too healthy. Okay? And you're also going to have to say, well, should we be talking bone marrow transplant or sickle cell? And I know we've got speakers coming up who are going to, talk more in that direction. But basically, you need to be having a fair amount of trouble with the sickle cell, because Health Canada and the FDA want us to be able to prove that this is making things better. So if you're already good, I can't show that you're getting better. Okay, so it's not that they're being mean. It's because it's our responsibility, and you also don't want a whole bunch of healthy people treated. Then you can say, well, I don't know if they went through all that. Was it really necessary? Kind of story. So that's the reason for these what's called eligibility or requirements for being on studies.
Thank you. And so we will get to some of the other questions in our question and answer period. So thank you again, Dr. Wall.
Thank you, Dr. Wall, and thank you, Robina. Our next speaker will be Dr. Greg Gilcher. Dr. Gilcher is the clinical lead for pediatric hematopoietic stem cell transplant at the Alberta Children's Hospital, University of Calgary. He holds leadership roles within the Sickle Cell Transplant Advocacy and Research Alliance, known as STAR, and the principal investigator for Project Sickle Cell Cure, which is a prospective registry to study the quality of cure for patients undergoing hematopoietic stem cell transplant for sickle cell disease. His career goal is to facilitate access to curative therapies for sickle cell disease in sub-Saharan Africa. Welcome, Dr. Gilcher.
Thank you, Dr. Mburu. Now, I believe I can share my screen. Is that correct?
Yes.
And can you see my slide okay? Great. Well, thank you very much for joining today, and to the organizers for the invitation to present. I'm going to walk you through what a bone marrow transplant or stem cell transplant or hematopoietic cell transplant, whatever name you put to it. There are various names that we put to this procedure. So explain to you what it is and how it can cure sickle cell disease, but also why it isn't yet offered to everyone. And for many of the same reasons that Dr. Wall was saying, there are still things we need to learn about how to make it safer. And certainly our goal is to offer safe cure to everyone. That's our dream, but we're not there yet. So we'll talk about what the risks are and why some people might be offered this possibility and others might not be. And there's a lot of activity happening in Canada, and I want to go through some of the advances in care and research in this country, and then we'll have the opportunity to answer some of your questions. So I'm going to try to make, there we go. I have one conflict of interest to disclose. So a biotech company has supported some of the STAR research efforts. So I just want to talk about what might someone expect from a cure, because there's a tremendous amount of hope that comes with this word and applies to gene therapy as well. And for us, at least in 2021, cure means no new damage from sickle cell disease to the body. So preventing new strokes or any strokes from happening if a child has never or patient has never had a stroke, and to prevent ongoing injury to the vital organs in their body. We think, but this is something we're still learning about, that damage that is already done is probably not reversible. And we hope that no new damage will happen after the transplant, but these are things that need to be studied further. And importantly, we want to improve the quality of life. We want less pain and ideally no pain. We're learning more about patients who've had chronic pain syndromes going into transplant, and whether or not that chronic pain experience resolves completely, or certainly we've had experiences where that takes quite a bit of time to resolve, if it will resolve. And this feeds into discussions about what the best time to offer this therapy is. So we need to learn more about not just the cure, but the quality of the cure, and hoping that people with sickle cell disease can have a more normal life. What is hematopoietic cell transplantation? I mentioned this can be, all these different terms really are referring to the same procedure. And a lot of people think of it as a surgery, but it's not a surgery. There's some medicine or radiation or immune therapy or a combination of those things that are given to a person. And it really is a real journey, a procedure that involves a commitment of months or years through its entire course. So it is the procedure of taking blood stem cells from a donor into a recipient. And as Dr. Wall presented, gene therapy in its current application is essentially what we call an autologous transplant, where the donor and the recipient are the same. There's chemotherapy given as gene therapy is currently delivered. Of course, you've heard about goals to change how that might look in the future. But in this case, the donor is someone else, so it's not the recipient. So these blood stem cells are taken from another person, typically a family member and typically a fully matched sibling, as I'll describe to you. And they're infused into the person with sickle cell disease after they receive about a week of some sort of therapy, which will weaken or deplete their immune system to some degree so that their body will accept these new blood stem cells. Because if we just gave them the blood stem cells, at least in 2021, without any sort of what we call conditioning or preparation, their body would just reject those new blood stem cells from the donor right away. And the donor could be a family member, which is by far and away the most common situation for sickle cell disease due to the safety and the best outcomes with a family member, a fully matched family member, a sibling, speaking of specifically. But there are studies ongoing looking at whether or not this could be more safely done with someone outside the family or a half-matched family member. And I'll mention and describe this in the coming slides. So the way this can actually work for a person with sickle cell disease is the blood stem cells from a donor are given to the person through an intravenous infusion. It's not a surgery. It's remarkable that this can be done through an intravenous infusion. I'll show you a picture in a second describing this. And these blood stem cells have the capacity to grow up into various types of mature blood cells, such as white blood cells, which protect us from infection, platelets, which help us not bleed, and then red blood cells. And so these blood stem cells from a person without sickle cell disease will grow up and produce normal donut-shaped red blood cells that do not sickle. And so in picture format, we have a donor who can donate these blood stem cells either by being put under anesthesia and having multiple needles put in their bone marrow into the pelvic bone, and pulled out and sent to the laboratory. Or depending on the way the transplant is done, and there are different ways to do it, these blood stem cells can also be collected from the bloodstream of the donor using a machine the same machine that would be used for automated red cell infusion or exchanges, but with different settings so that the stem cells are collected. And they go to the laboratory for processing. And meanwhile, the person undergoing the transplant receives some combination of either chemotherapy or radiation or immune therapy, and there are different ways to do this. And then the blood stem cells are given back to them through a special intravenous line. And that special line is in place not because it's specifically required for the infusion. A peripheral IV can be used, but because we need those special lines to safely support patients through this experience, because there's much more than simply giving them the medicines for a week before and then giving them the stem cell infusion. The recovery period thereafter is quite complex, and it varies depending on the type of transplant, although things are changing such that we're looking at even doing some types of transplants as an outpatient, but we're not there yet. So a lot of people want to know, why isn't BMT offered to everyone? Well, certainly it has risks, and it depends on what the donor options are. It depends on what type of conditioning that I described earlier is given. And importantly, sickle cell treatments are changing. So we have, fortunately, more advances in sickle cell care than we've seen in decades. And as new therapies are available for sickle cell disease, including newer transplant approaches, including gene therapy, and including disease-modifying drugs, we have to balance the benefits and risks of each of these therapies against each other, and look at an individual and see how their sickle cell disease is affecting their life and which one might be the most appropriate. The safest donor is a fully matched sibling, which is only available in less than 20% of people with sickle cell disease. So while the results are very good with a fully matched sibling donor, most people don't have this option. And to really summarize this scenario for most people who do not have such a match in their family, there are these other options that we're studying, such as half-matched parents typically, or sometimes half-matched siblings, or donors outside the family, but these are still considered experimental, though the field is changing. And of course, the perception of and perspective of the patient and family are paramount here. What is acceptable risk? That certainly must be a conversation that is shared. And the experiences and the expertise of the care providers is important to consider. Everyone wants to do the best thing for those patients we care for with sickle cell disease, and no one wants to harm anyone and doesn't want to make things worse in an effort to try to cure them of their disease. So I mentioned the best choice is a fully matched sibling, and I'll mention that transplant for sickle cell disease goes back to 1984, and that was a patient who was transplanted for leukemia who happened to have sickle cell disease as well. And there have been over 2,000 transplants done worldwide, and the rate of increase has really picked up over the last 10 years. And this is because there are much safer options than were available previously, with fewer short-term and long-term side effects. But the vast majority of these transplants, I mentioned about 2,000, about 85 to 90% of those transplants are with a matched sibling donor. And still 2,000 transplants is a high number, but compared to the number of people with sickle cell disease globally is still a very small number of patients. So a matched sibling donor transplant, depending on the center and their comfort level and the perspectives of families and the physicians at those sites, might be offered as routine care or as part of a research study. But it's fair to say that for a match outside the family, someone on one of the registries being the donor, is typically only offered as part of a research study or really under exceptional circumstances. And one of the points to emphasize with unrelated donors is, even if our research gets us to the point where using a donor outside the family, if we could achieve the same results as we can currently describe for a matched sibling donor, it doesn't solve the problem of access to cure, because people of African ancestry, which is not everyone with sickle cell disease, but most people with sickle cell disease, have very diverse immune proteins, and finding a match on the registry is quite a challenge.
Therapy for sickle cell.
So I wonder if everyone could mute their microphones, please. Thank you. So where is this treatment offered in Canada? So all 6 of the Canadian pediatric transplant programs have delivered this therapy at some point. And a few of the adult programs have done so. Our adult program in Calgary has done a few young adult transplants, and I believe Princess Margaret and possibly one of the Montreal adult hospitals may have done so as well. And certainly there's increasing interest as the safety of offering this therapy to young adults becomes more clear with more research. But the eligibility depends on a lot of factors. People have to be comfortable that this therapy can be delivered safely, and people are always wanting to do the best for their children or their family member, or in the case of care providers, they want to do the best for their patient. And as I mentioned, the vast majority of transplants are with a fully matched sibling donor. And I won't go through a list of all the research that has been done describing the outcomes with a matched sibling donor, but to simplify, the cure rates exceed 90%. Some studies even as high as 95%, and the risk of death, which is certainly not zero, so this is why we are not dismissive of these risks by any means, is considered to be about 5% or less. And we've learned over time that the outcomes are better and the risks are fewer when the patient is younger, but the challenge with this, this reality is that the disease may not be severe at that point of a child's life. And so either families or providers might not be comfortable recommending this treatment if the disease is not severe at that point in their life. But we know that the outcomes in terms of not only survival, but the risks of having major complications during the transplant are fewer when the patient is younger. So future goals, we want to reduce this risk even further. We're never satisfied with what we think are pretty good numbers, but we want zero risk of death, and we want higher rates of cure. And we also want to reduce risks of complications such as infertility, which understandably is a side effect of some of the older ways we've done transplant with high doses of chemotherapy that have been unacceptable to many patients and families. So I want to highlight some of the research activity that's happening. Dr. Mburu mentioned that we're part of the Sickle Cell Transplant Advocacy and Research Alliance, and most of the Canadian programs are now part of this research network. And one of the studies that we have available, and this is open at Sick Kids as well as at our institution, is an effort to reduce toxicity with a matched sibling donor undergoing transplant. The rates of graft versus host disease, which is a potentially life-threatening and certainly a potentially life-limiting complication of transplant, is much lower with this protocol when it's been studied in adults. Rates that that have been pretty close to zero, which has been a huge advancement in the field. And we've, based on some pilot data that our institution has produced, this is now being studied to make sure we really understand the risks and benefits in a multi-center way. And one of our key goals is to look at fertility preservation, and there's a signal. We don't understand this fully, but there's a signal between information from the National Institutes of Health and then some patients at our site and Children's National Medical Center that that fertility preservation is possible, but we don't really understand the percentages yet. There's another study called we call it the early transplant trial, but it's looking at if transplant is done earlier, can we improve quality of life in these younger patients, do this safely with a fully matched sibling donor again, and also protect their brains in terms of neurocognitive function when the cure is achieved at a younger age, and to look at their quality of life and their overall health and the functioning of the various organs in their body. So this is going open already in the United States, but the University of Manitoba and UBC in Vancouver are planning to open this study. So what about half-match transplants? So this could potentially solve the donor option or the donor availability problem that I described earlier, where most people don't have a matched sibling donor, a fully matched sibling donor, and most people with sickle cell disease can't find a fully matched donor on the registry, even if that was a safe option for everyone, which it is not yet a safe option for everyone. So if a half-matched parent or sibling could safely donate and we could have excellent outcomes, then this could significantly expand access to cure, because most people will have a half-matched parent or sibling available. And historically, this has not been offered to everyone because of high rates of that complication I mentioned called graft versus host disease, or the patient rejecting the graft called graft rejection. But this is an evolving field. Newer techniques are making it safer. And the literature is really growing. So we are excited about this, but there's still more that needs to be learned before we offer it to everyone. At one of our big meetings in December, there was a signal that maybe younger patients under 18 might be more prone to rejecting their graft. So we have to figure out what can we do to fix that and make this not only work well, but be safe and offer more accessible cure to everyone. So there's a huge opportunity for research here. So other projects to summarize, Mathoni mentioned Project Sickle Cure, which I'm leading. And so this is really a study looking at the quality of cure. We want to look at what are the long-term neurologic and neurocognitive outcomes and the pain and patient-reported quality of life after transplant. And this will be offered at many Canadian centers. We're hoping, this is still a work in progress, to make haploidentical transplant clinical trials more available, but that's probably going to take a little bit longer. Young adult transplants. So we're having discussions monthly with the National Institutes of Health and STAR, looking at ways to make transplant safer and more effective for young adults, particularly more effective because the transplant protocol that they've developed at the NIH is quite safe. It just has about a 15% chance of graft rejection. So we're trying to see if we can improve upon that. And then the hope is that this might be something that several of the adult transplant programs in Canada might be interested in participating in, but that's still in the early stages. We're part also of a long-term outcome study led by Emory University. We really want to look at how patients are describing their quality of life across various domains long-term, and also looking at their long-term cardiovascular health and fertility as well. We've amassed a large number of patients, 300, with very detailed data in a retrospective registry within STAR, and so we really have to start looking at this data and describing better some of the other outcomes of transplant that have not really been well described up until now. So we have a lot of the studies describe whether patients survive or not, whether they have recurrence of their sickle cell disease or not, and whether they have certain transplant complications. But we don't, unfortunately, despite those over 2,000 transplants that I've mentioned that have been performed internationally, have enough information about how the kidneys function long-term with or without a transplant, or lung function, as just examples. And we have some encouraging pilot data within STAR to suggest that a newer medicine might help prevent that complication, graft versus host disease, for unrelated donors, and that perhaps this could become a safer option for more people, but this needs to be studied further. And as I mentioned before, we are hopeful that this will expand access to safe transplant for more patients, but it's not going to solve, it's not going to allow everyone to access cure, because not everyone can even find an unrelated donor on the registry that matches with them. So we still have a lot to learn. We want to learn more about long-term brain function and the function of other organs, quality of life, pain outcomes, and how patients and families view this experience. So how could groups such as yours help with access to safe cure? Well, if we were to look at the partnerships between transplant and pediatric hematology oncology programs with childhood cancer organizations, we have made tremendous advances through partnerships with our community advocates and patients and families. And as I'm sure everyone knows on this call, that there's a reason and a history that has led to distrust due to poor relationships in the past, but really the way forward is through partnership and through partnership and research, and including patients and families in the discussions about what research priorities are important, and to help advance our knowledge about how to safely care for people with sickle cell disease, and ideally how to safely cure people with sickle cell disease. And also to note, there is a huge disparity in access to financial or support resources or supports for patients undergoing transplant who have non-malignant conditions compared to those who have cancer. And we struggle in providing these supports, such as housing or resources to cover medicines or food or transportation, because many patients will have to relocate to one of the transplant programs to access these kinds of therapies. And there's just not enough in the way of resources to help these families. And this is where childhood cancer charities and community groups have helped children with cancer, and there's an opportunity here for patient advocacy groups for sickle cell disease to help as well. So in conclusion, sickle cell disease can be cured, but matched sibling donor transplants, which are available to less than 20% of patients with sickle cell disease, is really the safest option and the only option routinely offered, depending on the center, outside of a research study. And I hope you've seen that Canadian hospitals and universities are becoming leaders in the field. We need to work with our patient and advocacy groups to help guide our research priorities and to facilitate access, with the ultimate goal of having a safe cure available to everyone. So thank you.
And thank you, Dr. Gilcher, for a comprehensive and excellent presentation. In the interest of time, we'll address questions in the Q&A period, but we'll move on to our next speaker, who is Dr. Gaurav Korea. So Dr. Korea is a senior consultant in pediatric hematology, oncology, and immunology, and a hematopoietic stem cell transplant specialist at the Apollo Hospital in Delhi, India. He has extensive experience in bone marrow transplantation for sickle cell disease and has pioneered protocols for haploidentical transplant for sickle cell disease with excellent success rates. He's also led initiatives to build curative therapies for sickle cell disease in India and other resource-limited countries, including several in Africa. So we're very happy to have Dr. Korea joining us this morning from India, where it's actually nighttime, where he must be exceedingly busy, as we've had insight into the recent and terrible surge of COVID-19 in that country. Today, Dr. Korea will be speaking to us about advances in stem cell transplantation in India as an example of a low, medium resource country. Thank you, Dr. Korea.
Good evening, good morning, everyone. So as rightly mentioned, it's almost close to 10 here. So thank you very much for having me on this platform. And I'm sorry I had certain challenges, but I tried to manage these 20 minutes. I'm having some emergency at home, but I'll try to be with you as long as possible. So the topic allocated to me today for discussion is advances in HSCT, hematopoietic stem cell transplant for sickle cell disease in India, an example for LMIC. And my previous speaker, Dr. Greg, has actually set a very nice platform for me, because he has already covered the basics of transplant and what has been done so far as the transplant for sickle cell disease is concerned. So my focus will be more towards what we have done in our country, how we have sort of improvised our strategy to meet the needs of patients who come to us. The brief overview of my presentation would include a few case scenarios. So each of these cases, what I feel contains has a message to carry, and subsequently we'll be briefly looking at the experience from India, evolution of transplant for sickle cell disease, evolution of novel strategies, what we have been doing in India. Then we'll briefly talk about gene therapy, gene editing. Dr. Walls has already talked about it, so we'll touch base what we are doing in India as far as gene editing or gene therapy is concerned, and subsequently we'll move on to the Q&A session. And I have no disclosures to make as far as my talk is concerned. So when we talk about hemoglobinopathies, there are two phenotypes which primarily come to our mind. One is the sickle phenotype, and the other one is the thalassemia phenotype. So these are the two most common prevalent hemoglobinopathies across the globe. And when we talk about the management or the treatment options for these hemoglobinopathies, we can broadly divide into two categories. One is the care, which is a routine care. So these are chronic illnesses which starts right from the time of birth, and they are there with the patient till end of the life. So they need so they are more of chronic ailments. So we broadly categorize the care into the treatment options into care and the cure. Care, we all know. So care, or we all know, includes medicines, some medicines which need to continue for good quite time, or for lifelong. These medicine needs to continue, some vaccination, other lifestyle modifications. And when we talk about cure, as of now, as Dr. Greg has also described, the only curative option is hematopoietic stem cell transplant, as we await a wider application of gene therapy or gene editing for these disorders. Now, why is hematopoietic stem cell transplant a more realistic option for lower middle income countries? So we know that there is a huge burden of the disease. So this is a disease where majority of burden is in resource limited countries. Okay, it's a chronic ailment, as I have described, and most of the newer drugs which are now available, which have added a lot of therapeutic options for sickle cell disease patients. Unfortunately, these new drugs are far out of reach of these patients. Okay, the access of these patients, these medicines, is also a challenge in resource limited countries. So HSCT is one-time treatment option. Offers a one-time cure. It's more cost-effective when we compare the lifelong expense of supportive care versus a one-time curative option. Yes, it has its own risk and benefit, very nicely highlighted by Dr. Greg. But over the years, the outcomes have improved by a better understanding of the transplant physician of the disease and continuous efforts to modify or develop strategies to tackle the challenges, whether it's graft versus host disease or graft failure. So we'll briefly have a look at all a few case scenarios. So all these pictures which I have put, they have been put here with the permission of the parents. So this is Master T, 10-month-old resident of Uganda. He was symptomatic since early infancy. So by the time he reached us, he had approximately 2 to 3 admissions with significant complications. So he underwent. So he was started on hydroxyurea and other supportive medicines quite early in life. By the time he reached us, he was approximately 1.5 to 2 years of age, and he was not responding to hydroxyurea optimal doses of hydroxyurea, and he was still not responding appropriately. So we screened the patient, and fortunately we found a sibling to be HLA identical. We discussed with the family, and we reached to a conclusion that transplant will be the best option, and this is the perfect time to offer transplant to this child. So he underwent transplant, and it's more than 5 years post bone marrow transplant, and this kid is doing extremely well. Another girl, 6-year-old girl, resident of Zambia. Now, she was well till 3 years of age, no significant concerns, mild crisis, but from 3 years onwards till the time she reached us, she had back-to-back strokes, leading to significant neurological compromise. Now, when she reached us, we screened the siblings to be for HLA matching. And one of the sister was found to be HLA identical. We discussed with the family that yes, we are offering transplant. So the intent is to prevent any further damage, but whatever damage has happened, it's difficult to say that to what extent this damage could be corrected. So we took a conscious decision and we took her up for transplant. She's free of sickle cell disease now, almost, I would say, 4 years post bone marrow transplant. But the question arises that what about the damage which has already happened to this girl? Was this damage preventable if she would have been offered transplant relatively early in life before these significant life-limiting complications happened? Moving on to another girl, 11-year-old girl, resident of Uganda. She was asymptomatic when she came to us, but very vigilant parents and a regular monitoring of transcranial Dopplers, and she was well till around 7-8 years of age, but around 9.5 years, they noticed that the transcranial Doppler velocities, which has, one of the speakers have discussed the importance of transcranial Dopplers. So they noticed, they were told that the transcranial Dopplers are now going into the range which puts her at high risk of having a stroke. So this was the time when they were told that if we can consider her for transplant as a therapeutic, as a curative option, it will be good for her. Again, we screened the family members. There's two siblings. So one of the siblings was found to be HLA identical. And we took her up for transplant, and she's also around 5 years post bone marrow transplant, doing extremely well. So this case is in contrast to the previous case, where early intervention and timely intervention actually helped us to compromise the quality of life for this girl. Now, this 19-year-old girl, a very pathetic quality of life, frequent transfusion, frequent crisis, miserable quality of life, frequent absenteeism from school. Now, it's very clear, based on all the indications available for transplant, she is a strong candidate for transplant. So when she came to us, we screened the family. She had 5 siblings, but unfortunately, none of the siblings was found to be a complete HLA identical match. So she underwent a haploidentical transplant using one of the sibling as donor. And she's more than 5 years post bone marrow transplant. She's doing extremely well. I think she's doing her master's now somewhere in China. This 14-year-old girl, a resident of central India, symptomatic from 6 months of age. But unfortunately, she was not diagnosed. She was only diagnosed at 5 years of age. Thereafter, she had multiple challenges. She was on hydroxyurea, optimal supportive care by one of our pediatric colleagues. But unfortunately, despite the optimal supportive care, she was not responding appropriately. So she met us, and we counseled them about the option of transplant. We screened the family members, but her sibling was not found to be a HLA identical. So we did an extended unrelated donor search, and fortunately, we found one of few unrelated donors for her, and we took her up for unrelated donor transplant. So this is a old slide. Now she's more than a year post transplant, and again she's doing extremely well, living a pain-free life. So with these cases, we move on to the disease burden. So we know that sickle cell disease is a huge problem across the globe, especially in the lower middle income countries. Millions of people across the globe are affected. People living in sub-Saharan regions, Africa, African Americans, Middle East, and India, etc. Approximately 75 to 85% of the cases are in Africa, and the projected numbers are going to increase by till 2050, and that is the time when they will actually start plateauing. Approximately 6 to 9 million kids are born with sickle cell disease each year in Africa. There is no definitive data available from India, but from some smaller studies, the disease frequency is as high as 1%. Now, what are the current therapies for management of sickle cell disease? This has already been covered by the previous speakers. So broadly, we can categorize them into supportive care, which includes penicillin, vaccination, pain control medicines, chronic transfusion, iron chelation, and hydroxyurea. When we talk about the curative options, the options we have is transplant. It could be myeloablative or reduced intensity. Again, this has been very nicely covered by my previous speaker, Dr. Gray. Now, moving on to Indian experience. So how the transplant has evolved in India. So the first time the transplant happened in the country was somewhere in 1986. So from 1986 to 2006, over 20 years, we can see in this slide that hardly there were around 10 or 15 centers across the country who were offering transplant. And if you look at the number of transplants which are done in these 20 years, hardly approximately 1,000 or 1,200 transplants were done over a period of 20 years. And if we look at the indications of these transplants, so these two bars, so the black bar shows the autologous transplants and the white bar shows allogenic transplants. When we look at the distribution of allogenic transplants, predominantly they can be divided into thalassemia and non-thalassemia transplants. So the number of sickle cell transplants were actually dismal. There were hardly any sickle cell disease transplants which was done. So overall, the number of transplants were also less, and the sickle cell disease transplants were also extremely less. Now, that was 2006, and today we are in 2021. A lot has changed in these past 10, 15 years. And what has changed? The number of dedicated centers which are now offering transplants have increased from those 15 centers to more than 100 centers across the country. Number of doctors with interest in pediatric hemato-oncology and transplants have increased significantly. There's the introduction of formal training programs across the country. There's timely referral of patients needing bone marrow transplant, and a lot of my colleagues have been to actually Sickle and they have got trained there, and they have come back and they have practiced and they have established excellent, fantastic transplant centers across the country. There is increased public awareness, better supportive care. There's liaison with international donor registries now, such as German registries, DKMS, NMDP, Anthony Nolan, so various registries. And more importantly, now there are some new Indian registries which have come up, like Datri, IDMR, or Bharat, which has increased the local donor pool for matched unrelated donors. So how has this helped us? So because of this, all these changes, number of transplants have almost quadrupled. The outcomes of transplants have increased significantly because more and more trained people are now offering transplant. Mud transplant have also gone up considerably with good success rate, because now the number of patients who have available donor, matched donor, has increased because the number of registries have come up. And the success of haploidentical transplants have increased tremendously due to better understanding of transplant immunology and the modification of strategies. Moving on to transplant for sickle cell disease. So we all know this, the first case report of cure of sickle cell disease, which was published in NEJM in 1996. So this was a patient suffering from acute myeloid leukemia, which simultaneously got cured of sickle cell disease when she was offered bone marrow transplant for acute myeloid leukemia. Now, again, this is old data, and Dr. Greg has presented an updated data already. So this was till 2013 when close to 1,250 transplants were offered for sickle cell disease. Okay, so very small number of transplants. And as Dr. Greg highlighted, currently there are around 2,000 transplants which have been offered for sickle cell disease. So HSCT for sickle cell disease is an unmet need. Why? Because more than 140,000 patients with sickle cell disease are there in Europe, USA, and more in emerging countries. Approximately 40,000 patients had indications and an HLA-identical donor in Europe and USA, but they were not offered transplant because of one or other challenges. If we include the African continent, Middle East, and India to this number, then we can imagine that these numbers of patients who actually need transplant is countless. What is the status of transplant currently globally? So each year, approximately 100,000 transplants are offered, maybe more. My colleagues might correct me if I'm wrong. Amidst this, only 1,200 transplants were offered till 2013. And if we take the updated data as highlighted by Dr. Greg, it's around 2,000 transplants. So we can see the contrast: 100,000 transplants overall every year, and 2,000 transplants overall for sickle cell disease till date. So why is this difference? What are the factors responsible? What are the obstacles with sickle cell disease? So few of them have already been highlighted by Dr. Craig. My understanding is that there is lack of clear-cut indications for transplant. So majority of the physicians who are dealing with these patients across Africa, India, Middle East, they don't have a clear-cut idea that which patients are candidates for transplant. There's lack of awareness within the physicians. There is lack of awareness within the parents about the outcomes, improved outcomes of transplant. So the fear of transplant, which is inherent there in the physicians as well as the parents, that is still persisting. Lack of funds. So finances are an important aspect. Okay, so the cost of transplant is significantly high. So we have done a lot of effort to bring down this cost, and somehow we have managed to a significant extent, and the cost of transplants have come down to close to $25,000 to $35,000 for these patients. And then lack of available donors, as highlighted by Dr. Gray, the chances of finding a HLA-identical donor within the family is very less. Less than 20% of the patients have HLA-identical donors. So what will happen to another 80% patients who actually need transplant? So brief about my journey of transplant for sickle cell disease. So this was the first child whom I transplanted when I came back from UK after getting trained at St. Mary's Hospital, Imperial College. under my mentor Josu. So he was a 5-year-old child, significant sickle-related comorbidities. So he underwent a matched sibling donor transplant. It was one of the most challenging transplants I've done. He was on ventilator with acute chest syndrome at the time of transplant. So not ventilator, sorry, he was on BiPAP support. We did the entire transplant on assisted ventilation, and he did pretty well. So we can see the date of transplant, and now he's almost 7 years post-transplant, doing very well. So we talked about the indications. So the indications have been evolving over past 2 to 3 decades. And this is the latest set of indication used by Bolanos et al., which was published in 2012. So this has expanded the indication. So if we go through these indications, we'll feel that majority of patients who are having challenges, complications with sickle cell disease meet one of these criteria and are might be candidates of transplant. Now, what is our experience, Indian experience of transplant for sickle cell disease? So this was a data which we presented in EPMT 2018. So overall, around 73 transplants were done across 6 to 7 centers. Majority of them were matched sibling donor transplants. 2 of them were matched family donor, 3 were matched unrelated donor, 2 cord blood transplants, and 15 haploidentical transplants. So out of these, 17 median age was 9 years, male to female ratio was 45:28. Majority of the patients were from African Union or Middle East. I'll not go into the dosages and other complexities of the transplants. I'll straightforward move on to the outcomes. So if you look at the outcome of these transplants, the overall survival was 89%, and the disease-free survival was 83.5%, and the transplant-related mortality was 11%. So this was since the time transplant started, so this includes over experience of almost 15 years or so. So why is there a need of alternate donors? Okay, so we know that less than 20% of the patients have HLA-identical sibling donors. So what will happen to the 80% who actually need transplant but they don't have HLA-identical donor? So HLA-identical unrelated or haploidentical donor are other options which can be considered. Now, talking about the HLA-identical unrelated donors, due to the poor representation of voluntary donors, especially from African descent in the registry, the chances of finding a matched related donor is extremely, extremely poor. So this brings this HLA mismatch or HLA haploidentical donors in picture. What is the advantage? The main advantage of haploidentical donor is that you always have multiple donor available in the family. So even if you're a small family where one child is affected and you have 3 potential donors, 1 sibling and 2 parents, and you can screen all of them, and whosoever is found to be best as per your selection criteria, he or she can be used for transplant. So this was the first haploidentical transplant which I offered after coming back to India, which was in September 2014. So this child is again 7 years post bone marrow transplant, and he's doing extremely well back in his native country. So this was the first modification which we made. This was not actually a modification. This was the same strategy which I was using at Imperial College, where I was trained. So this was the pre-transplant preparation starting from day -60 with hydroxyurea, azathioprine, and hypertransfusion. This was the basis was the original paper by Bolanos, but Josu modified this strategy and added this pre-transplant preparation to it. BMT was done using this conditioning. So I'll not go into the intricacies, but it is sort of a reduced toxicity conditioning. With this strategy, our overall survival was around 80%, and the disease-free survival was 71%. So not very exciting. So it was different to what I saw in London while I was getting trained. So then I realized that these are the different set of patients which we are getting. So the patients whom we were treating, they were very different. They were very well managed. So the graft failure rates were very less. So, and the patients whom we were treating here, they were in a different shape. So that is why the graft failure rates are quite high. So we modified our strategy and we added some more bit of pre-transplant preparation by adding hydroxyurea, by adding sort of fludarabine and dexamethasone to hydroxyurea as a thiopurine. The BMT conditioning was the same, and the post-BMT follow-up was also the same. And with this, we achieved some success with the overall survival of around 89% and a disease-free survival of 80%, but still we were not there. So we made a third modification, which we called as the POLO protocol. And this is what we did. So we added 2 cycles of flutarabine, cyclophosphamide, and dexamethasone as pre-transplant preparation to sort of mitigate the risk of both graft failure as well as the rejection. Okay, and this was subsequently followed by this was subsequently followed by a bone marrow transplant. One additional thing was we used upfront plerixafor for mobilization of the graft, and we used a post-transplant cyclophosphamide, sero, and MMF for graft versus host disease prophylaxis. So,
sorry, Dr. Karia, just a 1-minute warning. I apologize because we're running short on time.
Sure, no problem. So with this, we managed to achieve some significant outcomes, and which can be highlighted here. So the engraftment was very, very robust, and we can see that the outcomes were close to 90% for haploidentical donors, which was a success for any resource-limited country. The graft versus host disease acute as well as chronic rates were also less than 20%. Again, there is more. There is a possibility of decreasing it further, and we are striving for it. But this was again a very significant thing which we managed to achieve. We moved on to the MASH unrelated donor setting using the same strategy, and we experienced that the outcome of the same strategy in MASH unrelated donor setting is also pretty good, with very minimal or absolutely negligible risk of graft failure and graft versus host disease. So our overall experience so far, we have done close to 900 transplants. Demographics, 2 are from India, rest are from Africa and Middle East. If we talk about the overall survival, it's around 86.73%. Overall survival, if we divide it into last 2.5 to 3 years versus prior, so the outcomes are definitely much better in past 2.5 to 3 years. But the disappointing part for me as a transplant physician in the country is that just 2 patients are from our country. So whom to transplant and when? So this is a quote from Shalini Shenoy's paper, a review article, which says that as in all non-malignant disorders, for the best outcomes, transplant for sickle cell disease should be performed when a recipient is at a good functional baseline in anticipation of the inevitable organ damage that is driven by the duration of pathology and thus the age. So I'll skip these slides about the gene therapy and gene editing because Dr. Walls has already covered it. But we are also trying trying our hand on gene editing for hemoglobinopathies, and this is sort of a rough workflow of what we are trying to do. Let's see where do we reach, and possibly whenever we interact next time, probably I'll be having something more to present as far as gene editing strategy is concerned. So with this, I would like to conclude my talk with few take-home messages that sickle cell disease is a preventable disease. As soon as the diagnosis is made, if unfortunately after taking all the precautions, if it's still a child is born with sickle cell disease, as soon as the diagnosis is made, which is usually in the first year of life, supportive treatment should be started as soon as possible. A close observation is required to identify severe phenotypes. BMT should be offered to these severe phenotypes as soon as possible, and a long-term follow-up is should be ensured. And as we make BMT more amenable to the masses, we should also prepare ourselves for future modalities such as gene therapy or gene editing. I would like to thank you very much for a patient listening, and I'm happy to take any further questions. Thank you very much.
Thank you so much, Dr. Karia. And so that was another excellent presentation. We are very spoiled this morning. And so I'll introduce our final speaker, and then we'll have a Q&A period that will be open to everyone. So I'm very happy to present Dr. Kevin Kuo, who I've known since we started our hematology training as subspecialty residents in Toronto. And so Dr. Kuo is an adult hematologist and clinician investigator in the Red Blood Cell Disorders program at Toronto General Hospital, UHN, and he's also an assistant professor in the Division of Hematology at University of Toronto. His clinical focus is on rare congenital hemolytic anemias, and his research focuses on the impact of sickle cell disease on neurocognition, novel therapies in sickle cell disease and other rare anemias, and non-chelating methods to treat and prevent iron overload. Today, you'll be speaking to us on the subject of newly approved therapies and how we can best utilize them in view of current therapies that are in development. So thank you, Dr. Kuo.
Thank you, Evie, for the very generous introduction. So today my topic of my talk is decisions, altering the treatment paradigm in sickle cell disease. These are my conflict of interest disclosures. I receive an award from the National Institutes of Health, NHLBI. I receive grants from Thalassemia Foundation Canada, Peter Munk Cardiac Centre, University of Toronto, Cincinnati Children's Hospital Medical Centre, and the Canadian Hematology Society, as well as Pfizer. I perform consultancy for Agios, Alexion, Apellis, Arovent, Bluebird Bio, Celgene, BMS, Novartis, and Pfizer in alphabetical order. I'm a chair of the Data Safety Monitoring Board for a zinc finger gene editing study in sickle cell disease for BioVeratif and Sanofi. And I have research collaborations with Phoenicia Biosciences and Apfero. So today's objective is to review newly approved therapies in other jurisdictions, to explore their mechanisms of action and clinical outcomes, and discuss how these treatment paradigms can be altered as a result of having these new therapies. Usually, most of you know that I usually talk about clinical trials that are in action right now, especially the ones that are in Toronto. But today, I think I want to step back and I want to sort of talk about what's sort of closer to the horizon as opposed to, you know, the therapies that may be 5 to 10 years away. And as many of you know, despite the fact that we have had many of these advances in the treatment of sickle cell disease, including gene therapy and stem cell transplant, that there's still not a lot of options at the moment in clinic. And mostly when we see a complication in someone with sickle cell, we say, is there any evidence-based treatment option? If there is, we would initiate it. And usually the evidence would point to hydroxyurea. And if there's clinical improvement, great, we continue on hydroxyurea. If there's no clinical improvement on hydroxyurea, you know, we move on to transfusion. And if transfusion doesn't work, then we say, oh, they're a match-related donor because we can perhaps pursue a bone marrow transplant. But if not, then we usually end up in the realm of clinical trials. Now, as I have talked to you for many years already, that there's a number of therapies that were in development or are in development, and many of them have gone through the gauntlet of clinical trials. But it's important to remember that of 100 new treatments that are being pushed from the laboratory into clinical trials, only 7 of them ever make it through this gauntlet of phase 1, 2, and 3 clinical trials, because A, the clinical trials need to prove that these drugs are safe. And by and large, they're mostly pretty safe because we have had vigorous testing in the animals already in the preclinical phase. But more importantly, we want to prove that these drugs can perform the vital role of showing clinical efficacy, not just a change in some hematological parameters, but to actually make a difference in someone's lives. I mean, that's the most important thing about taking a drug. And if a medication cannot prove that, then there's no point in taking it now, is there? And this is why it takes such a long time to develop a therapy, and it takes such a long time to and it's so risky to actually develop a therapy. So of all the therapies that you see here, you know, that are in blue, in gold, and in gray, the ones that are in blue and gray, the ones in blue are currently in clinical trials. The ones in gray are the ones that have been tested in the past but have failed to show any benefit in patients. And so really all we're left with these days are just 4 therapies. And you'll see that not all 4 therapies are approved in all jurisdictions, and I'll show you why. We have hydroxyurea, we have voxelotor, we have crizanlizumab, and we have L-glutamine. So we look at the ones that are approved right now. Hydroxyurea, yes, is approved in both United States and in EMA. EMA is the European Medical Agency in Europe, and by Health Canada. You can see that FDA approved hydroxyurea way back in 1967 and in the pediatric population in 2017. For in the European sphere, the hydroxyurea was approved in 2003, and Health Canada also was very similar to FDA. This drug has been approved for many, many years now. The three new ones I'm going to talk about are L-glutamine, voxelotor, and crizanlizumab. L-glutamine was approved by the FDA in 2017, July. However, the company withdrawn its application to the European Medical Agency in 2019, and they have not filed any application for approval to Health Canada. Voxelotor similarly was approved by the FDA in 2019, but they're planning, quote unquote, planning to submit in middle of 2021, and there's no application to Canada yet. Crizanlizumab is approved by both the FDA and EMA in 2019 and 2020, respectively. Again, they have no applications to Canada yet. Now, remember that the approval of drugs is not initiated by the regulatory agency. It's not that Canada doesn't want to approve these drugs. It's that the company needs to submit an application for the drug to be approved in order for it to approve. And then you're going to ask me why, well, how come companies don't necessarily, if they've gotten drugs approved in the US or in Europe, why don't they come and file it in Canada as well? Well, A, filing costs money because it's a lot of regulatory hoopla. B, is that the market may not be large enough for these companies to sustain these operations, and so they don't necessarily file with us. And C, sometimes their focus could be elsewhere. But there are ways to mitigate that, as I'll show you later. So let's start with hydroxyurea. Hydroxyurea has a number of effects. It can increase the amount of blood that are being produced in the bone marrow. It can reduce the stickiness of the red cells and the stickiness of the white cells onto the capillaries and onto the blood vessels, so that it can improve oxygenation. It can also increase a globin called the fetal hemoglobin, which is a baby hemoglobin, which carries oxygen very well, unlike the sickle hemoglobin. And it can also donate nitric oxide, which is a very potent dilator of blood vessels, which again improves the flow of blood. And you can see that the hydroxyurea really was, in sickle cell disease in particular, was developed through a very, very long time, from 1984 all the way to 2010, with a number of clinical trials that have been done. And you can see that unequivocally, hydroxyurea has improved pain crises. And the lines that I see here, there's a line through the middle, and then any dashes that are to the left means that they're in favor of hydroxyurea. You can see that it improves pain crises, it improves it reduces blood transfusions, it reduces hospitalization, it reduces acute chest syndrome, can reduce hepatic sequestration. There is a good chance of reducing stroke, as well as we talked about transfusion as well, and splenic sequestration as well. So going back, so now that we have talked about hydroxyurea, and hydroxyurea, of course, has a number of biases against it, such that a lot of patients are not willing to take it because of, either because of the misunderstanding of how the drug works, or the, perpetuation of myth, including, that it is it's a drug that can cause cancer. And because of a whole host of factors, a number of companies have devoted themselves to, and also researchers have devoted themselves to developing new therapies. So of these three new therapies, let's talk about the L-glutamine. So why did MS, the company that makes L-glutamine, withdraw? Officially, the company says they withdrew because of a change in company's marketing strategy in May 2019. But really what happened was that the EMA actually have decided to refuse marketing authorization based on the following. They said, this is the European Medical Agency said, the main study did not show that Zindari, which is the trade name for the L-glutamine, did not show that it was effective at reducing number of pain crises and hospital visits, and a large number of patients that were taking the Zindari dropped out of the study before it was finished, and they considered the way that the data of these patients that were dealt with was not appropriate. So what do they exactly mean by that? Well, what is Endari, first of all? We don't exactly know what the mechanism is. We don't know how exactly it works, but we know that L-glutamine is needed for the synthesis of NAD, which is a sort of a battery in red cells. And the uptakes are several times greater in sickle cells than compared to normal. And we know that L-glutamine is associated with, increase in these power batteries called NAD at least in sickle cells. And we know that if we treat sickle cells outside of the body for 4 weeks with L-glutamine, that it can, reduce its stickiness to blood vessels compared to the controls. But how does it really work in humans? Well, this company, MS, did a phase 3 study. They had 230 patients with sickle cell disease. They claimed that the L-glutamine significantly reduced pain crises compared to placebo, 3 versus 4 pain crises, and there were fewer hospitalizations, 2 as opposed to 3. But if you look carefully, and here I have reprinted the FDA briefing document on L-glutamine, on Endari, you can see that there were a considerable non-ignorable number of patients who dropped out of the study. In fact, 36% of patients dropped out of the study before completing the study by 1 year, compared to only a quarter of the patients taking placebo. So clearly, whatever side effects that L-glutamine gives is not acceptable to these patients. Nonetheless, the company says, oh, well, you know what? These patients dropped out. That's okay. What we're going to do then is going to take the results of the patients who remain in the study and then apply to those who dropped out. Now, it somehow makes sense to the company, but it doesn't make sense to me, because if you dropped out of the study, you're not going to be benefiting from the drug. So how can you assume that the benefit is derived, is the same as those who remained in the study? But nonetheless, that's what they did. So the FDA says, no, we don't like that idea. We're going to reanalyze your data. And when they reanalyze the data, really, there's not much of a difference between those who are taking placebo versus those who are taking L-glutamine. They have a quote-unquote hazard ratio of 0.73. What that means is that 1 is like no difference, completely no difference. Anything less than 1 is that, it shows probably a slight favor to L-glutamine, and anything higher than 1 is showing favor to placebo. You can see that, yes, it shows favor in favor of L-glutamine, but the confidence interval, meaning that how confident can you be with this result, really is touching 1, meaning that for all we know, this result could be just chance and there's really no difference. And then the European Medical Agency did the same analysis again, all over again, and they also found that there was no difference. And more importantly, the number of pain crises that were reduced were, you can almost say they're not that significant, because the number of pain crises counts reduced from 3.8 in the placebo group down to 3. So elimination of about 0.8, like not even 1 crisis, just 0.8 of a crisis in 1 year. Now the FDA at the end of the day said, well, you know what? We're still going to approve you, not because we think that your drug is particularly effective, but because it's a nutritional supplement and we think it's generally safe. This is why it was approved. But the EMA says, you're not showing any benefit and we're not going to approve you. Stop there. So that's why they withdrew. So this could also be the reason why MS has not filed any application in Health Canada for approval. Now on to the next drug, voxelotor. This is an interesting drug, because what it does is that it binds to the interface between the alpha and beta globin. In fact, it binds to the alpha globin, but it sticks to it permanently. And what it does is that it can alter the flexing of the hemoglobin such that it's more accepting of oxygen. And you can imagine that if a sickle hemoglobin is more accepting of oxygen, then its ability, you sort of reduce its ability to sickle, which is great. The problem with voxelotor so far is that all the results they published are based on laboratory outcomes. Well, no one's pain crisis is ever going to improve based on laboratory outcomes, right? And you can see that only 1 abstract that was published by Vachinsky had change in vasoconstriction crises as an outcome. There was no reduction in painful vaso-occlusive crises compared to placebo, but there was only a trend towards reduction in the cumulative incidence of vaso-occlusive crisis when we stratify by hemoglobin value. So to me, that's a bit uncertain in terms of whether this drug will work to improve clinical outcome. It certainly improves laboratory outcome, but there's no improvement in clinical outcome. So the FDA granted accelerated approval, but it's conditional on completion of the pediatric and adult trials. And here I list all the trials that they're supposed to complete. Furthermore, they're required to present clinically meaningful results. The problem here, though, is that the trials began in May 2016, but they have not presented any data yet. And that's concerning to me personally. After you run the trial for 5 years and you still haven't generated any, clinically meaningful data, it's like, how long do you need to take in order to show that it's clinically meaningful? Last drug is crizanlizumab. There was a phase 3 SUSTAIN study that was published in 2017. There was a huge celebration with this because this was a study for a very, very long time since hydroxyurea to show that there is an actual, almost you can say a meaningful reduction in pain crises. So they have shown that the median number of pain crises were reduced from 2.98 down to 1.63, so almost by half. And they have also shown that the time to the first pain crisis was significantly lengthened if you were taking a drug. Instead of 1 month to crisis after starting the drug, your time to first crisis after starting the drug was 4 months. And the time to second crisis was also longer. Instead of 5 months, it's 10 months, et cetera, et cetera. The problem, though, is that the initial manufacturer of crizanlizumab, which was initially called Celg1, ran out of drug. They had a very small production facility. They were using Chinese hamster ovary cells to make the medication. And it's because they ran out of drug, they sold the rights to Novartis, and Novartis has to remake the entire manufacturing process. So instead of making it out of Chinese hamster ovary cells, they're now making the drug out of slugs, which is apparently more effective. And sorry, you get a high yield. So great. The thing, though, is that because you now change manufacturing methods, FDA now requires them to reassess the entire process. So they have to look at whether the drug can create antibodies in the body, which will neutralize its effect. Because if its effect is neutralized, then you won't be able to use it. It also needs to look at things like infusion reactions, infections, and bleeding. So they have to redo basically everything. And we're currently waiting for the results of this because the trials just got started. So going back to this initial roadmap that I've shown everybody, we have all these promising drugs, but most of them in blue that are still in development, that are not even close to being approved yet. The ones that are in gray are completely gone. We're really left with only 4 options. So where do we go here? If these drugs will be available to us, right, in Canada, is it one silver bullet? Or do we strike multiple targets simultaneously? Meaning that do we use combination of these drugs? Already, some of these trials have been aimed at sort of looking at this combination. The trial with crizanlizumab was in combination of hydroxyurea, meaning that you can be on hydroxyurea. So some people were actually on hydroxyurea and crizanlizumab at the same time. Same as the voxelotor. Some patients were already on hydroxyurea and they were allowed to remain on hydroxyurea. Is this going to be the way of the future where we combine maybe crizanlizumab with voxelotor, or maybe voxelotor within a new drug, or crizanlizumab within a new drug? That is waiting to be seen. But what I want to do is sort of plant this question in your brain so that when we come to discussion, that we can start talking about this in a sort of an informed and meaningful manner. And then the question is when to strike. We know that, for example, stem cell transplant is much more effective and in children compared to adults. Then perhaps in children we should use stem cell transplant, and then we should reserve these therapies, newer therapies, for those who are older, perhaps. I think the factors to consider in the treatment of sickle cell disease is that we now have a number of high risk, high reward therapies like gene therapy and stem cell transplant, which can provide cure. But then, there's still a role for these traditional disease modifiers, given you may or may not know, recently there's been sort of 2 AML cases, leukemia cases in patients who have undergone gene therapy, the Atlantic globin virus gene therapy by Bluebird Bio, and those trials are now put on hold. So because there's still a huge gap between the development and also when the product is actually matured to be put out on the market, I feel like there's still a huge role for disease modifier to be considered for our patients. Disease modifiers like drug therapy. That's what I'm trying to talk about. The problem with drug therapies is that we can control sickle cell, but we can't cure it, right? That's the downside. So factors that I think we need to consider are things like the inclusion criteria. Right now with gene therapy and bone marrow transplant, the inclusion criteria is quite limited for many people. So they may not be able to benefit from one of these therapies. When you think about accessibilities to these new drugs, right? The companies are not even filing the application in Canada. So how are we supposed to even get access to these drugs to begin with? Are we trading one complication, over another? And then also we have to consider the idea of early adopters, right? For some people who are willing to take risks, they will consider clinical trials. But then for others who are more conservative, they'll naturally want something to be approved before coming out. It's also important to think how we're gonna, fund these drugs, right? There's several levels of intervention we can have. We can advocate for ourselves in CADF, which is an agency that looks at what we call health technology assessment, or assess whether it's worth it to fund the drug or not, based on various evidence that we have, clinical trials. PCPA is where the pricing is set. It's negotiated, I should say, between the provinces and the drug companies, and also, of course, provincial public formularies like ODB in Ontario and Pharmacare BC, or in Alberta Drug Care. We are now working towards building a national strategy for high-cost drugs for rare diseases. Hopefully, this will encourage companies to bring their new therapies into Canada. And with this, I draw to a close and I welcome any questions. Thank you.
Great. Thank you very much, Dr. Kuo, for that comprehensive talk on newly approved therapies. I believe what we are seeing with Health Canada is there's a lot of barriers and there's a lot of red tape that needs to be overcome for these therapies to be made available. Thank you. That brings us to the conclusion of our webinar, and now we are moving to the Q&A session. And I believe you'll all agree with me. We have been fortunate to have this wide range of speakers for this very engaging session, that we are now asking you to give us an extra 10 to 15 minutes to be able to go through the questions that have been posed by the group. I will just share my screen and we shall go through the questions that was posed for each speaker. And so to start with, for Dr. Odame, I don't know if he's still online with us.
Yes, I am.
Thank you. What are your thoughts? This was asked by Abby. What are your thoughts about elevating our political advocacy? It seems to me that it seems to me that we need to yield the big stick. What are we all doing about political advocacy in our communities?
Yes, I think what we ought to know is that there are other rare diseases like metabolic disorders, other neurodegenerative disorders, very rare diseases that have very expensive treatments, that are also struggling with having access to new medications. And rather than going it alone, I think the sickle cell community should really join the fight for all rare disorders. There are strong advocates like Dorhane, like John Adams, who sits on the board of Sickle, I think all these individuals should bring their skill set and link up sickle cell disease with other rare disease organizations, because the numbers of patients, if we take the whole of Canada, is small. And often that is a barrier to you having a voice. So I think we should band with all these entities and going together to reduce the red tape that goes on, maybe simplify the whole process of approval so that we'll be able to get access to this drug. As Dr. Cole mentioned, I don't think these new drugs will be for everyone. And I think there should be a reasonable sort of guidance as to who will benefit from it. But the clinicians also need a voice in it so that in the end, patients will benefit from these new treatments.
Thank you for that response. The next question was by P.A. Bayliss. Are these drugs approved for children in the countries where approval exists? I believe she was referring to the newer agents, Volexator and crizanlizumab.
So they first get approved in adults. Actually, I was on the Data Safety Monitoring Board for Voxelotor's development. And currently pediatric trials are going on, and the same with crizanlizumab, where pediatric trials are going on. So once the approval is first done in adults, it then moves down to no different from the way COVID vaccines are being developed. It's usually developed in adults, and then gradually the age is dropped. And as more studies are done, they become approved for children. So I think we have a bit of a time now to really develop a strategy as advocates and make our voice heard. And I think the way to do it is to join forces with other advocates who are advocating for a policy in Canada that accelerates the process of getting access to some of these men. We understand that cost implications are important, and we are responsible citizens, but at the same time, we want to advocate that the interests of the patients should be the driver for all these negotiations.
Okay, thank you. And Samuel asked, could you please repeat the factors that make these agents more efficacious?
Well, I think as you've heard, many during these clinical trials, there were patients who were taking hydroxyurea. Hydroxyurea is really a winner. And today in Canada, there are too many patients with sickle cell disease who should be on hydroxyurea and are not. I think it's a battle that we have to fight to make sure everyone is on. But hydroxyurea is not a cure, and there are some patients who continue to have pain, even though they're on hydroxyurea. I think these patients will benefit from crizanlizumab. Or there are patients who have low hemoglobin, they have a lot of breakdown of red cells, and Voxelotor has shown efficacy in improving the hemoglobin and in breaking down, reducing the breakdown of red cells because it's an anti-sickling agent. So I think the efficacy was demonstrated even in patients who were taking hydroxyurea. So I think a combination of therapies is probably going to be the way to go. There's not going to be one magic medicine that will take care of everything. And so we should have access to these drugs, and clinicians with patients together have to decide what combination of therapies, either one or combination, will be able to help them.
And the final one by Beverly, I believe, was a comment. She said, my son is on hydroxyurea and he's doing well. I was just concerned of him being on it for life.
Well, you saw, I mean, I think being on it for life, I think more and more we're seeing patients who've been on hydroxyurea for nearly 25 to 30 years. We haven't seen any signals yet. But also, I think when we come to these costs and curative treatments, I'm not lost on, when I show you the cost of the drugs, some of these drugs in the US now, I mean, if you're going to treat somebody $100,000 a year, every year on a medication, then probably a one-time curative therapy might be more cost effective. So I think some of these things will change the way we look at curative therapies, Because if we're going to have yearly annual treatment in the hundreds of thousands of dollars, then it makes sense that one-time curative therapy might be a more cost-effective approach. So I think all these considerations will come in. These drugs are efficacious in addition to hydroxyurea or without hydroxyurea, and that's the way we have to look at them.
Okay, thank you. I will let my co-moderator take the next set of questions.
Okay, thank you, Dr. Mburu. And I guess there's one question in the chat that nicely kind of links into these questions here about hydroxyurea. So Cynthia is asking, could Dr. Kuo please provide more details on available data that speak to the effects of hydroxyurea and fertility? What are some things patients can do to reduce these effects?
Thank you for the question. There is currently great uncertainty as to the effect of hydroxyurea on fertility. And the reason for that is because we know that even without hydroxyurea, many patients with sickle cell disease have reduced fertility. This is also confounded by the fact that many people who are on hydroxyurea are able to successfully conceive, despite having lower sperm count. If we're dealing with a sperm number of 1 million plus, reduction by 50% may seem significant, but then it's still a lot. Remember, you only need one sperm to fertilize an egg. And so because of these very, very confusing comments, I think personally, I think it's even more confusing than what the messaging of COVID-19 these days is in terms of whether hydroxyurea causes fertility issues or not. From my personal experience in my clinic, none of my patients have had issues with having children while on hydroxyurea, and they're all adults. So in fact, I think sometimes they may be a bit too successful at it. And so I think the important thing right now is to, discuss the options with your clinician and make an informed decision as to, as to what treatment is best for you.
Thank you. And there's another question here in the chat for Dr. Odami. What are your patients who are on hydroxyurea? Sorry, what about your patients who are on hydroxyurea and still having frequent hospitalization and pain crises? What else can be done for these patients who are in pain every single day without missing doses of hydroxyurea? When will these other treatments be offered to these patients?
Yeah, this would be the ideal group of patients that you want to add monthly infusions of crizanlizumab, for example. And I think that's where crizanlizumab actually would be a big addition to the treatment. People who are taking hydroxyurea, they are adhering to the treatment, they've had some reduction in pain episodes, which is significant, but still not sufficient in terms of minimizing pain and improving their quality of life. So I believe that we are going to go into combination therapy, and there are many people who enroll in the crizanlizumab trial who continue taking the hydroxyurea and still saw extra benefit from taking the crizanlizumab. So these are going to be some of the indications we will have for crizanlizumab when it gets approved in Canada.
And if I may add to Dr. Odami's comment, because many of these companies have not filed for application for approval in Canada, many of our centers are engaging in clinical trials using these medications so that we can offer our patients access to these new medications, even if they're not available in Canada. For example, at the Toronto General Hospital, we have both the voxelotor clinical trial as well as crizanlizumab clinical trial.
Thank you. And then I have a pair of questions for Drs. Wall and Dr. Correa, as Dr. Correa didn't have a chance to talk about gene therapy, but he had referred to it. So the first question is about the risks related to the infusion of the gene therapy agent and whether or not chemotherapy and radiation would be involved. And then the second question or comment is about a desire for these therapies to be available in low and middle income countries. So what are some of the barriers for that, or has this been done in India, for example?
So gene therapy or gene editing, as we all know, so it's sort of it's form of autologous transplant. Okay, so it's a manipulation of the autologous stem cells, but subsequently the process of transplantation remains the same. So autologous transplant also, irrespective of whatever is the disease under consideration, it involves administration of chemotherapy. Okay, so this strategy also has chemotherapy, although the agents which are used are different. The combination of agents are different, because a very important component of transplant, allogenic transplant, is a serotherapy, which is not required here. Okay, the intensity of conditioning is also a little bit less. So predominantly majority of the conditioning regimens so far include busulfan. Okay, so it is almost the same with minor modifications. So this was, I think, the first question, whether the gene editing or gene therapy incorporates the use of chemotherapy or not. So yes, it does. Secondly, what are the complications? So the complications can be they can be manifold, and we all should understand and realize that it's an evolving field. You might have heard of a recent news which came up because of which this Bluebird trial has been suspended, because around 3 patients who underwent gene therapy developed acute myeloid leukemia or myelodysplastic syndrome sometime after undergoing a gene therapy. So the trial has been suspended, and they are trying to figure out what was the reason. Most likely, the reason for myelodysplasia or acute myeloid leukemia was because of the increased predisposition of sickle stem cells to have these changes, with the added effect of busulfan. So it's less likely that it was a contribution from the gene therapy, but that still remains investigation, and probably in another 5 to 6 months we'll have more clear-cut answer. So that is about gene therapy. Another strategy is gene editing, which is CRISPR-mediated gene editing, where you very precisely cut the defective portion of the gene. So it is technically it is a more safer strategy, and I presented a slide about a recent publication by my friends and colleagues Selim, Josu, of gene editing in patient with sickle cell disease and thalassemia, with excellent, very encouraging outcome. So this is about the gene therapy and gene editing. What have we done in India? So it's too early as far as gene therapy or editing is concerned in India. But yes, we are taking some baby steps. We have actually conceptualized what we plan to do. So our strategy is a gene editing based strategy, where we intend to harness the effect of hereditary persistence of fetal hemoglobin. We all know that if the fetal hemoglobin percentage is beyond a certain range, these patients are less symptomatic. So we are trying to optimize the fetal hemoglobin modification and bring it to the optimal level. But still, it is in laboratory, and we have to do animal trials, and subsequently we'll apply for clinical trials. But yes, if we are able to to show significant results, the cost will be very, very different from the cost which is currently being offered for either gene therapy or gene editing across the globe.
Thank you, Dr. Karia, for your response. Dr. Wall, would you like to add anything to that?
Yeah, so basically, it's what I had shown you in my talk with the current results with the gene editing, and you know, as things are evolving, you know, yeah, you know, we are basically in the first phase of things. Ideally, in the next generations, we will be using, we will be able to target the therapy without having to pull the cells out of the body, without having to come in with the chemotherapy. So that's our goal. Short term, we're using the tools of bone marrow transplant, but it is significantly less chemotherapy than what we use in a bone marrow transplant. The combination of chemotherapy and immunotherapy compared to what we use in a bone marrow transplant, but there is no long-term data. Okay, and so this is all exciting. We're still in early stages, and looking forward to to moving the field forward.
Thank you for that response. There was 2 other questions for bone marrow transplant. Dr. Gilcher had to sign off. What is the minimum age for BMT patient and donor? Second question is, how can patients outside Canada access bone marrow transplant in Canada?
I can take the first question because I think Dr. Greg is not here. So as far as the age is concerned, basically for any disease, when we figure out that yes, this is a patient, this is a candidate for transplant, then I think that's the right age. Okay, and as I highlighted in one of my concluding slides, that the transplant should be offered early in life to get the best possible outcome and best possible quality of life. Okay. So suppose a child is diagnosed at 6 to 8 months, we try to optimize our supportive care, hydroxyurea and other stuff. We see what the effects are, and if this kid is not doing well on optimal supportive care, then we should consider for transplant. There are very nicely laid guidelines now, indications for transplant, which are very freely available. One of that was highlighted in my slides. And if the child continues to be symptomatic on optimal supportive care, he or she should be considered for transplant as soon as possible. Preferred age is anytime after 2 years, but yes, we have done transplants even before that. Second question was how a person out of Canada can access transplant in Canada. I think Dr. Walls or someone else in the forum might be able to answer it in a better way in absence of Greg.
Yeah, no. So we do transplants as well, but so the other part of the question was donors. How young for a donor? We have to pay attention to the safety of the donor, and so usually we wait till the donor is at least 6 months of age, depending on the size of the recipient. The limitation for the patient being treated is not age, but it's the having time to show us that this is going to be a patient who has severe sickle cell anemia. Canada is a publicly funded healthcare system, so as such, we're not set up to take international patients, and so we don't have a medical tourism infrastructure. We're just not set up to do that, and I'm sorry to tell you that.
Great. I believe we're coming to the close of all our question session. The last question is regarding COVID. We have recently gotten letters to book our teenage patients to go for the COVID vaccination. Is there any information on how young people are responding to the vaccine, and more so the sickle cell disease patients in Canada? Anyone can take it.
Yeah. Yes. Get your vaccine as soon as it's available. If you're a caregiver and can get the vaccine, get it. If you are, as it's moving down, get it. Dr. Simpson, I just was on a call that, there's each regional health authority even difference within cities has different rules. And so we're trying hard to standardize. But right now, as of this week, we're vaccinating down to 16. The responses are beautiful in the 12, between 12 and 16 age range. I expect school vaccinations will start soon. And there's no reason to think, that there's going to be a lower end on this. And we'll, the plan is to, as soon as we can get vaccine, to roll it out. I don't know, Dr. Simpson, if you've got a more updated from CHIO.
Right. Yeah. Thank you. Yes, I would echo your thoughts there. And, here at CHIO, we have started to immunize caregivers where the parents or other caregivers of children who are 16 and over right now with sickle cell disease are also eligible. And so we are trying to encourage our families to take this opportunity because, these are safe and effective vaccines and our best chance of keeping ourselves well, avoiding the severe complications that can happen with COVID-19 and sickle cell disease, and hopefully overcoming this pandemic. But I encourage everyone that if you have questions or if you have concerns about it, that you reach out to your sickle cell team so that we can guide you through it and help you to access the vaccine if you don't know how to access it. But like Dr. Wall was saying, we strongly encourage you that when you do have that opportunity, you take it. Okay. And so thank you, everyone. I think that Launry had a comment that she wanted to make. She had her hand up. I see she did post the link for the video. Thank you. That's great. Launry, did you want to say something, or was there anything you wanted to add?
Yes, hello. Can you hear me?
Yes, we can.
Okay. I just wanted to thank all the speakers again. I think I am echoing. Sorry about that. And also to mention that it's been a pleasure to have all of you on this session today. With respect to Dr. Odami's comment around advocacy, I wanted to mention that Duran Wangrega is also on this call today as an organization. We are working with the community to ensure that we do have, life-saving drugs and therapies, coming to our country. So for sure, this is something that we will continue to work on. With the COVID-19 vaccination, as Dr. Donawal advised, please get your vaccine. We actually fought to get sickle cell listed in phase 2 for the rollout. So we are there. So please take advantage and get your vaccine on time. Again, I want to apologize that we went over the time, but you will all agree this is a great session with excellent speakers. And so we thank you again for being part of this session today. Thank you. And I'll hand it over to the great moderators again to conclude the day. Thank you.
Thank you. Thank you again, Launry. And so that brings us to a close today for our webinar. And so this has truly been an inspirational session with an outstanding group of speakers. So we're very grateful. Thank you so much to Drs. Odami, Dr. Wall, Dr. Gilcher, Dr. Correa, Dr. Kuo for sharing your time, your expertise and passion this morning. We're clearly in an era of hope when it comes to the therapies that are being developed for the treatment of sickle cell disease. And so I'd like to thank all of our attendees for being with us today and for your questions and the great discussion we had. I hope that you're leaving this session with renewed optimism for not only the future of sickle cell disease, but potentially your own personal future, the future of your family members, and the future of our patients for our healthcare providers here. Last but not least, I would like to thank SCIGO and Ms. Launry Tunji-Ajayi for making this event possible and for inviting Dr. Mburu and I to moderate this session. So everyone, please stay well, keep distance, stay safe, get vaccinated as soon as you can. Okay, thank you everyone.